chose Navigating the Challenges of Pharmacogenomics Implementation and Clinical Practice. So the CPPI is about moving pharmacy practice forward. So I tried to design a talk that would speak to the audience and the core of those that are served by the CPPI. It's the VCU policy to share the code for the CE credit in the beginning and at the end of the presentation. So you all can take note of the code here. It expires in 48 hours, so don't miss out on your opportunity to get that hour of CE. And I have nothing to disclose. I have no financial connections to any pharmacogenomic companies or any producers of genomic testing kits or any of those types of things. So I'm sharing hopefully unbiased materials today that are free from commercial bias. So our objectives today, we'll review the principles of pharmacogenomics. Hopefully at the end of this talk, you'll be able to utilize pharmacogenomic biomarkers that are currently available to personalize pharmacotherapy and use educational resources that are available for pharmacists and healthcare practitioners. And then towards the end of the talk, and somewhat interwoven in between, you'll see what are some of the barriers for moving pharmacogenomics broadly into pharmacy practice. And so what I don't show you on the objectives is the fact that we're going to start with how did I get here? So what made me fall in love with pharmacogenomics? So why do the principles of pharmacogenomics speak to me as a person? And you all get to cheat sheet here. This is background information here. This is my vision board from elementary school. So parents keep everything, and my mother kept this in a folder with all kinds of documents, my good documents and the bad ones, you know. So she kept these things, and central to my identity at the time was not that I was just cool, smart, or whatever, competitor, an athlete, right? Headed toward the Olympics or the NFL, you know, something like that. But I said that I was a craftsman, right? And so how did I end up becoming a craftsman? So craftsmen, like you have to be comfortable with using tools, making measurements, getting things right. Like I was a craftsman. And how could I make that claim as an elementary school student? Because I worked for my dad, and my dad was a cabinet maker. We made custom cabinets to fit homes. They had to fit perfectly. They had to meet the specifications of the homeowner, or we were not going to get paid. And so I once saw a customer make some request, or he was slightly unsatisfied, and it was the floor's fault. His floor was running. It wasn't level in the house. But he was not going to pay my dad until we figured out how to make that cabinet sit flat, the base cabinet under where the sink is, so we made it fit. So we worked around it, used some tools, and made it look perfect for the homeowner. It taught me something about meeting the needs of those that we serve. So if we're looking at these sets of cabinets, this first set of cabinets here they're made from oak plywood. They're stained early American. That's a Minwax brand stain. I'm not selling Minwax, but I'm telling you, it was the specifics of making them fit the needs of the homeowner. And you see that they fit right up next to this window in the middle. The sink is lined up right in the middle. This is what they paid for. This is what they asked for. The color of the countertops are what they asked for, okay? These cabinets here in the middle, these are natural finished red oak cabinets. This is a green marble countertop. And you see this base cabinet has an L shape. Everything had to be perfect. We measure, we cut, we finish. This was how we did our work. And this set of cabinets on this side is later on when we had more advanced technology. These cabinets are natural finish maple with raised panel doors. So you see the little designing the door. This was fancy work. And there's an island in the middle. So there's space, right? We had to meet their needs for us to get paid. All right. This influenced how I would consider optimizing therapy for patients. Whatever tools we use, it's ultimately to get the outcome that we want. We're getting paid to do a job, or we're getting paid to help people be healthy. And we use tools to eliminate risk associated with therapies or to get the best possible outcomes associated with therapies. So this shaped who I would become before I ever landed at a university and, started to do research. So the clinical potential of pharmacogenomics is very similar to what I just described with making those custom cabinets. We're trying to get optimal outcomes for those that are suffering with chronic or acute illnesses that require medications. The FDA released this report paving the way for personalized medicine around 2011, and it was helping to demonstrate the need for pharmacogenomics as a part of their review process. And you can see that unacceptably high rates of or percentages of patients fail our gold line standard therapies for many chronic disorders. This was the point that they were trying to make. Pharmacogenomics offers this potential of being able to identify the best drugs for patients in a crowd. And this is the classic bubble people that I borrow from Julie Johnson. She originally used this illustration to describe pharmacogenomics in the early 2000s, 2001 or 2003. And this spoke to me coming from the cabinet shop, right? If we could identify people who a particular drug would be best for, we can give them that drug. Those that maybe that drug may not harm them, but it may not do them any good, we can give them something else. And then maybe there are some in a crowd that a particular drug would cause a serious adverse event that would cause them harm. We definitely want to give them something else. So the central dogma of molecular biology is that genes are ultimately transcribed to RNA, and that RNA is translated into proteins. So genetic variability in the DNA can ultimately impact proteins or the targets of drugs, the elimination routes of drugs, or transporters that influence the concentrations of drugs that make it into different parts of the body. Now, if we take a look at that, we'll take a closer look at the target of a drug. As an example, this is the beta adrenergic receptor. My mentor at the University of Florida at the time, Julie Johnson, who's now the director of the CCTR at Ohio State University. She's moved on. She was studying the beta adrenergic receptor. And the beta adrenergic receptor at that time, in the early 2000s, there were two polymorphisms or variants, one at codon 49 in this top part of the beta adrenergic receptor, and a polymorphism in the bottom at codon 389. These variants were associated with the function of the beta adrenergic receptor. So if the target of drugs like your atenolol, metoprolol, carvedilol, propranolol, the target of the drugs was variable, this could impact how well you were able to lower blood pressure. I'm going to show you that. But before I show you that, we're going to look at how important this particular codon 389 was. So this is a non-synonymous variant, or a variant that changes the actual structure of the receptor. And is this something that should make a difference? If you look across species, animals, humans, if you look at rats, mice, apes, dogs, cats, frogs, you see that this particular variant is evolutionary conserved. Across species, at that particular part of the beta adrenergic receptor, there's always an arginine. And when there is a glycine there by accident, or by mutation or variant, something happens, all right? So here in humans, if there's an arginine there, you have a robust response to a beta blocker. And I'm showing you data from metoprolol. And across different ways of evaluating blood pressure, systolic, diastolic, daytime systolic, or daytime diastolic, you see a big response when the receptor is functional. When the receptor is non-functional and you have a variant that is not normally there in humans, dogs, cats, frogs, all the way across, you lose that ability to impact blood pressure response. Now, that's just sort of a short-term phenotype, right? Or a short-term measure. What about long-term? Long-term, this is what you observe. So let's say we're using a beta blocker. In this case, beta blocker bucindolol was being investigated for treating heart failure. When they evaluated patients that were carriers of the wild type functional beta adrenergic receptor, you could benefit and reduce the risk of mortality in patients with heart failure. When you gave the bucindolol to patients that carried that variant glycine allele, you can see that there was no benefit with bucindolol over time. It was not any different than the placebo group, all right? So over time, having wild type function, you could keep patients out of the hospital, you could keep patients surviving longer, but when that receptor, the target of the drug was variable, you did not benefit those patients over time, all right? So variants would impact blood pressure response and survival and hospitalizations as an adverse event or adverse outcome in heart failure. Now, what about drug metabolism? Well, genetic variability can influence the functionality of our major elimination routes for drugs that we use in patients. And these variants have large impacts on not only the efficacy of medications, but it also impacts the rates of adverse events and serious adverse events. We'll take a look at some of this here coming right up now. So as an example, cytochrome P450-2C9 is the major enzyme for metabolizing S-warfarin, the active enantiomer of warfarin. Okay, so what happens when you have loss of function variants in CYP2C9 and you take doses of warfarin? You see these elevated plasma concentrations of patients that carry these variant alleles in CYP2C9. And it's pretty nice stair-step sort of looking data where if you have one variant, you're kind of in the middle and delayed elimination. But if you have two, you have this higher risk of experiencing bleeds and long-term concentrations of warfarin in the bloodstream. So genetic variability can influence the targets of drugs. It can influence the major elimination routes of drugs. And now I'll show you data on transporters. And so I'm going to focus on one of these transporters, OAT1B1. And so OAT1B1 is responsible for simvastatin going from the bloodstream into the hepatocytes where simvastatin does its work, but it is also where simvastatin is metabolized, okay? And so what happens when you have variation in this transporter that takes the drug from the bloodstream into the liver? Well, I used GWAS data to start this story. I knew I have some GWAS people in the room. And this was an unbiased approach, taking a look at one of the limiting side effects of statins, which is statin-induced myopathy. And so doing a GWAS, this unbiased scan of variants that impact or associate with the myopathy, this is what popped out. OAT1B1 variant associated with this increased risk of developing myopathies, and it happens fairly early in starting a high dose of simvastatin. And you can see that somewhere around the first month or 2 months, you already see this elevated risk in these variant allele carriers of OAT1B1 variants. Now, what about the plasma concentrations? I'm showing you the adverse event happening at high rates and significantly, but let's take a look at the plasma concentrations. If we look at this variant, the variant shown here and showing you where it impacts the structure of the channel. If you look at those variant allele carriers, having those homozygous variant status or the two variant CC alleles, you can see that your concentration of simvastatin acid in the plasma is about threefold higher than in the wild type. And this correlates beautifully with that increased risk of developing myopathies. So we put all of this together. If we examine the impact of genetic variability on the targets of drugs, on the transporters associated with concentrations of drug making it from main like bloodstream into different organ systems, or if we examine the impact of genetic variability on our major elimination routes of drugs, those metabolizing enzymes, we put all of that together, we get pharmacogenomics. 20 years ago, we were using the term pharmacogenetics, studying one base pair at a time, making assays in-house, one base pair at a time, spending years of your life to study 7 base pairs. That's what I did in my PhD work. I spent like 4.5 years studying 7 base pairs of DNA, right? And making essays, and then GWAS came about, right? So, but back on topic, when we consider now our broader approach and evaluate genetic variability in terms of targets of drugs and their elimination routes, we get pharmacogenomics. And we use that to predict whether or not we can control for variability in efficacy and toxicity in drugs. Okay, our first question pops up now. Genetic variation influences the responsiveness to medications via which of the following? And you all can answer in the chat. Anyone in the room want to throw out an answer? D, all of these. Great, you guys are doing great. We may clip the rest of the questions. You may test out early. You know, you are doing so good. Now, why are we talking about this today? A part of the reason that we are talking about this today, no longer do you have to spend 4.5 years studying 7 base pairs of DNA. Now, we have such great technology, and it is so much cheaper than it was like 20 years ago, that the cost of genomic testing is falling faster than predicted by Moore's law. And so I tell students that in the future, this is going to be free or completely covered by insurance, and we'll never know who paid for it, and we'll all have it. We'll all have our genomic results integrated at some point into our care decisions. And I believe that to be a fact. We're headed in that direction. Costs are falling so fast. Technology is improving so much that we now have broad access in many ways to clinically actionable genomic results. So for the audience of the CPPI, those that are influencing patient outcomes in their work every day, this is what's going to happen to us. This is a clip from a New York newspaper making a joke about a patient walking in with their genome and handing it to the community pharmacist, and he's there kind of looking confused, like, uh-oh, this is headed our way. So we have to get prepared for interpreting results that are coming. So I'm giving you some tools today, some available resources to help with understanding what is actually usable information and where to get resources to help with integrating this information into your clinical practices. I always recommend starting with the FDA in their Center for Drug Evaluation and Research. There's Office of Clinical Pharmacology. And in the Office of Clinical Pharmacology, there's a genomics and target therapy group. They publish a table that I'm going to show you the link to, where they put information, they gather the information on pharmacogenomics that are in the required labeling materials for approved drugs. So if this information is in the required labeling materials for drugs, that means pharmacists are responsible for it already. So you're already practicing pharmacogenomics against your will, maybe sometime, all right? And we're also going to review the CPIC, the Clinical Pharmacogenetics Implementation Consortium, which was originally funded by the NIH through the Pharmacogenomic Research Network. So at the FDA, currently, there are 609 pharmacogenomic biomarker drug relationship in the FDA-approved required labeling materials of prescription drugs. The package insert, when you're working in the pharmacy and you open a box and there's a folded pamphlet with all of the information about the drug, there's 609 drug gene pairs in those package inserts. 243 of those gene drug pairs are on classic or traditional pharmacokinetic pharmacodynamic genes. So the cytochrome P450s, your transporters, and the targets of drugs. These are the classic skill sets that differentiate pharmacists from other healthcare professionals. So the pharmacodynamics and the pharmacokinetics, primarily the pharmacist practices in that space or is the content area expert in that space. So that information is there. If you follow this link on the page, you can go and see the table for yourself or just Google it and you will find the table. This is what the page, the landing page looks like. And if you go there, look on the table, it'll give you an outline of the markers, where they reside in the package insert. And I'm just going to summarize that for you because we don't have time for a demonstration. To click on the link and get out of Zoom and then get back to the slides in the right view, right? We don't have time. So I'm going to show you this. If we examine just the pharmacokinetics data, you will see an overrepresentation of 2D6 variants, 2C19, 2C9, and then you see some relevant numbers of 2B6, 3A5, and 1A2 gene drug associations in the FDA materials. And what sections are they in the required labeling materials? The majority of it is in the clinical pharmacology section, which tells you how drugs work in people. So how they explaining the mechanism and elimination routes of the drugs in humans. So that's where the majority of these recommendations are. You have some that are dedicated to the adverse drug event section, some that are located within the drug interactions. Some of them are black box warnings, and I'm going to show you a black box warning, and some in the dosage administration, these different regions. Now, what types of drugs have this pharmacogenomics information already there, and you're practicing it, using it already? You see lots of important chronic diseases that are being represented by these categories. And I'll tell you, take a look at this. Psychiatry, overrepresented if we think about the total number of drugs, but they're overrepresented with pharmacogenomic information in the package labeling material that we are already responsible for. So also cardiology, neurology, you see what areas they are in. Broad, okay? Now let's take a quick look at the CPIC. The CPIC is a consortium that is comprised of experts across academia, from the industry, from governmental agencies, where this is a service. This is a labor of love in a sense. So they have full-time careers elsewhere, and they get together and write these guidelines out of the goodness of their heart, out of service to the profession. So because this is a service effort and not a dedicated paid effort, that means it's slower. There's some lag between when a guideline is written compared to when data becomes first available. So right now we only have 28 guidelines, but those 28 guidelines are touching important and impactful drugs. Um, if you go to the CPIC landing page, you can see what those 28 guidelines are, but they cover drugs like tricyclic antidepressants, SSRIs, opioids, um, drugs used in transplant, drugs used in neurology, NSAIDs, antibiotics like voriconazole, proton pump inhibitors, clopidogrel. So it's drugs that are commonly used and where variability makes a big difference on outcomes. Now, if we take a look at just the top 50 drugs of the top 300, if you go to the top 300 drugs, pull out the top 50 and start to look for guidelines, which ones of those top 50 drugs are impacted by CPIC guidelines? 40% of them have a CPIC guideline, the most commonly prescribed drugs. And we're talking about, if we add these up together, hundreds of millions of prescriptions total per year, where there's a guideline that's been written to help with optimizing that therapy. Now, these CPIC guidelines are written in a way to make it easy for non-clinician scientists to use the information. So they summarize it for you in an actionable way. They're not trying to make you experts on genotyping or calling variant allele, any of those types of things, okay? So question 2, who is responsible for establishing the pharmacogenomic materials that's in the required labeling materials of approved drugs? You all have options there on the screen. Those that are participating online, please drop it in the chat. And I see the correct answers flying in on the chat. So that is true. It's the FDA. So the FDA is non-biased and they don't have commercial interest in any of the information that they provide. I always go there, start with the FDA, because companies will come around selling you products that may not have evidence that is not influenced by their bottom line, right? So if I start I won't give you an example. If I start a company, you all might look at me bad afterwards, right? But ultimately, you make money in business. And so you might make claims to be able to predict outcomes for drugs that you don't have evidence for. Or you may extrapolate results to drugs that share metabolism cascades, and you may not have the evidence to support it. That happens. Just go to the FDA page and you at least cover it on the basics of what is acceptable evidence for pharmacogenomics. So what is the value of pharmacogenomics implementation? So I had the opportunity to sit in on a discussion that Ima Lohi was leading in the journal club with Dr. Salgado and the other outcomes faculty. And they were talking about these different elements of value. And I'm going to try to show you some different elements of value that are demonstrated in the data from pharmacogenomics data. And some of them that I will not show that have been observed are adherence improved, the value of hope. I'm going to show you insurance value coming up. There's been data showing around equity associated with pharmacogenomics, but many of these elements of value have been demonstrated, and I'm going to demonstrate some of them as we move right along. So what could be the value of adding pharmacogenomics to clinical practice? Well, if we take a look across data, examining adverse events, and I go straight to adverse events that are associated with hospitalizations here in my talk. you will see that there's an overrepresentation of drugs that have pharmacogenomics information in their required labeling materials or that have guidelines that have been written by CPIC that are associated with these increased rates of hospitalization. And if we go and examine readmission rates after a hospitalization, the same is true again. Drugs that have pharmacogenomics data in their required labeling materials or pharmacogenomic guidelines are associated with increased rates of readmission after discharge. So drugs that have variability associated with genetics contribute to adverse events associated with hospitalization and readmission. Now we'll take a look at, other than just hospitalizations and readmissions, what about long-term outcomes? A good example of this and demonstrating value is CYP2C19 and CYP2C19 and clopidogrel. So clopidogrel is a prodrug, and when we take it, it's not functional or it's not active against platelets, and it can't inhibit platelet aggregation. But once it's converted in the body to its active form, it can then go inhibit platelet aggregation. Well, the most impactful enzyme that influences platelet aggregation and activation of clopidogrel is CYP2C19. You all can see that CYP2C19 is variable based on ancestry or self-identified ancestry. And this starts to give you a clue that you're going to see variability in the responsiveness to clopidogrel long-term. Now, to take a look at this in the short term, CYP2C19 is shown to be the biggest influencer of the kinetic activation of clopidogrel. This is held true across loading doses of clopidogrel, whether it's 300 milligram loading dose, the 600 milligram loading dose, or the once daily maintenance dose of 75 milligrams. So across the bottom here, they're showing you phenotypes of CYP2C19, not showing you like the genetic star alleles or whatever, but saying poor metabolizers to ultra metabolizers or super functional or regular functional to poor going this way. And those that cannot activate the drug due to poor CYP2C19 function, you see that their kinetics mirrors their functionality of 2C19. What about their functionality? If you measure their mean platelet aggregation or the change in platelet aggregation, it pretty much follows closely the kinetic categories. So if you have non-functional 2C19 status, that's the largest impact on whether or not you're going to be able to limit platelet aggregation. So that's short-term kinetics, dynamics, or how the drug is working. But what happens in these long-term outcomes? In this same study, they show you long-term outcomes. And if you're not able to activate the drug, you have higher rates of poor outcomes, whether that is these composite primary efficacy outcomes or just examining closely stent thrombosis. So not being able to activate the drug is associated with poor outcomes in the long term. Now, that was replicated many times in this period between like 2008 to early 2010. This was very exciting. This was the talk of all of the conferences. Walking through the hallways, it was a buzz, like talking about, have you seen the clopidogrel data? It was like gossip. It was exciting, right? Very exciting. And it replicated many times. If you can't activate the drug, you get poor outcomes. It's like wasting money and setting people up for poor outcomes long term. What happened in March of 2010? The FDA put a black box warning on clopidogrel. The black box warning says clopidogrel depends on activation to an active metabolite, principally by CYP2C19. So it summarizes everything that I just showed you there in basically that first sentence. It says drug can't be activated without 2C19, and then it's associated with poor outcomes. And then they tell you to pick another drug if you have someone in that poor functioning category. So after that, in early 2013, a guideline was written. I'm showing you the update to that here on this screen, but you have experts, um, pharmacists, scientists, physicians that came together to write these guidelines from CPIC to tell us what to do with the information. And the take-home message from that guideline is this: if you have functional 2C19, whether that's normal function or hyperfunction, clopidogrel is okay to give to that patient. If you have somewhat diminished function of 2C19 or poor 2C19 function, consider an alternative drug. Take home message: this is what you can use in practice. You don't have to worry about all of the genotyping and all the different types of variants that show up, but they summarize it for us nicely and simply put here. So, question 3, and right now everyone has 100. This is a high stakes question. The black box warning that was added to clopidogrel is related to what? I know that you all know, so I'm just going to keep it moving, okay? Now, what about payers' perspective? Payers' perspective. So this is one of the exciting payer perspective stories that I've ever seen presented. I was at the University of Florida's pharmacogenomic meeting in 2023, and the speaker, the presenter, was a CPA, certified public accountant from the teachers retirement system of Kentucky. She was the presenter of the data, and it was fantastic. So this is their homepage on their website. So if you want to go to see their website, but this is the data that she presented. So they came up with a partnership with the life sciences company, and they engage members. They roughly have 30,000 members of the teachers retirement system in Kentucky, and they recruited volunteers to receive a pharmacogenetic test and then subsequent follow-up with a pharmacist to manage their medications based on the pharmacogenetic test. And so they ended up with about 5,000, a little over 5,000 that agreed to participate, and they matched those with the controls who did not participate in the pharmacogenomic study. These are their results. I'm summarizing them here for you. Those that received the pharmacogenomic test and management by a pharmacist had 14.9% reduction in inpatient hospital stays, 6.8% reduction in ER visits, 1.9% reduction in outpatient visits. They saved $218.34 per member per month in cost avoidances. Cumulative over the 32 months of the study, they saved $37 million. 3 out of 5 members had a prescription changed by a pharmacist. So 3 out of 5 of those that were genotyped had a prescription change. 100% of participants had at least one actionable pharmacogenomic variant on their profile. And about 66% had a pharmacogenomic gene-drug mix match, meaning that a suboptimal kind of combination was present. And that mirrored very closely this 3 out of 5 members having a change by the pharmacist. So their study was overwhelmingly positive for the payer. All right. Now I'll show you a little bit of data from justifying the value, a potential value here in Virginia. And this is data that my student, Richard Jerome Dixon, pulled from the Virginia All Payer Claims database. So the All Payer Claims database was established by, it's codified here in Virginia. And so it's publicly available. It's evaluating claims data from insurers so that you can ask questions in data and improve outcomes and the health of Virginians. So Jerome wrote this program in AWS. It's like an Amazon AWS, right? Big dashboard to pull data, right? From claims across 2016 to 2020, and we were pulling drugs that were initiated 45 days within someone then going into a hospitalization. So these drugs were started 45 days before someone ended up going to a hospital. And if we do take a look here, we'll see atorvastatin has a pharmacogenomic guideline that is written for it. Hydrocodone has a pharmacogenomic guideline that has been written for it. Omeprazole has a pharmacogenomic guideline, and oxycodone has a pharmacogenomic guideline. If we look across the data broadly, and we go further down into the list of drugs, we start to see many that have pharmacogenomic guidelines where these drugs were associated with hospitalizations within 45 days of starting these drugs. So we see beta blockers, we see opioids, we see antidepressants, multiple of them. It gives us a clue that maybe we can make a difference on outcomes and the economic sides of outcomes that are adverse associated with drugs here in Virginia. And if you want to see Jerome's landing page and you want to play around with the data and look at the data based on age of participants, it changes based on age. You can scan the QR code that he has created for you. And I can put this back up again at the end if you all want it, so I can make it through the slides, right? So what are some barriers to pharmacogenomic implementation? So I'm going to quote my department chair, not just because he's just smart, but I think he's right. He's smart and he's right in this case. So therapeutic inertia is impacting how we're moving pharmacogenomics forward. So Dr. Dixon was making this argument in terms of cardiovascular disease or in cardiology. I'm using the same argument for pharmacogenomics. So we have a lot to do in terms of clinician and patient education on what we're trying to get accomplished. And I'll later argue in the slides that one of our issues is with pharmacogenomics, it sounds like an academic research exercise when we're describing what we're attempting to do, when actually we just want to improve safety and efficacy outcomes for drugs, right? But pharmacogenomics sounds foreign. It sounds unfamiliar to those in practice. It sounds unfamiliar to patients. So we have to figure out a way to tell this story better so that we're not getting a bad reaction from providers and from patients. We also need teams dedicated to managing the safety and efficacy of drugs. And so this is something, the places that are doing large scale pharmacogenomics implementation, they have multiple faculty members. Like if it's an academic institution, they have multiple clinicians, faculty members, research scientists, lab members. They have big teams that are dedicated to solving some of these problems that we're going to see. And one of the major issues, finally, is the system approaches. How do we get the pharmacogenomic information and alerts into our clinical decision support software? And so one of the leaders in that space, the clinical decision support software space, wrote a paper about this, and he describes these challenges that are associated with the genomic information following a patient as they go from practice to practice or health system to health system. So even if we have the information available, how can it be accessible by the providers as this person moves along? How can we get the systems to talk? And if pharmacogenomic tests are eventually done at birth, as an example, like how does that information follow this person as they go on throughout their life? So these are some of the challenges, sharing the data and the data being accessible. I started this already saying that you know, um, pharmacogenomics is fun to me and exciting because I was a craftsman, right? That's what my story said. But we have to figure out a way to tell this story in a way that patients find synonymous to thermometers, glucometers, home blood pressure monitors, right? Like these are things that people are familiar with. We can sell them over the counter in the pharmacy. People come and buy it, and they don't even have to ask questions about it. They're pretty confident they're going to go home and check their blood pressure, right? At some point, these medication safety tests or dose optimization, whatever we want to say, we have to figure out a non-smart way of telling this story and connecting it to people. Okay. Uh, my summary and conclusions. Our current approach, or the past approach, has been apply pharmacogenomics after a problem has occurred due to some type of medication, an adverse drug event or adverse drug reaction. Someone is hospitalized, and then we try a test, and the test solves the problem or it gives us clues on why we observe what we observe. The future approach, and this will be better at some point when we have these results preemptively before prescriptions are written, and we can have alerts that help guide clinicians in their decision making, or that help a pharmacist understand what risks are added to a patient, similarly to those alerts that pop up now when you are in practice when there are drug-drug interactions. When there are severe ones, big alerts pop up and you have to override them to move forward to even fill a prescription. That's the direction that we are going towards in the future, and we'll have better outcomes associated with that. Now, I tried to save time for questions, but I also have some supplementary slides if someone else, it depends on what questions we get. But I ended at 12:50, so I hope that we have some time for questions.
Hi. So one thing that gets discussed a lot, I think more in like pop culture and media, is that patients are afraid that insurance companies will use their pharmacogenomic information to discriminate against them or to raise their prices. What are your thoughts on this, and how should we like counsel patients on this type of fear?
That's a great question, and I'm glad that you asked it. So in two in 2008, before leaving office, President George W. Bush signed the GINA Act. As it's like Genomic Information Non-Discrimination Act. So pharmacogenomic data cannot be used against you by insurance companies, by health insurance companies. It cannot be used against you. However, they excluded life insurance companies. Life insurance companies can use whatever they can gather against you to, to not have risk associated with high risk. Life insurance people can discriminate. Health insurance cannot discriminate using your genetic information. Great question. Yes, ma'am.
So what kind of time horizon do you anticipate for us to get to this point where you'll have pharmacogenetic information readily available for patient before you do the prescription?
So in some places it already exists. Like entire health systems have preemptive genotyping. As an example, at the St. Jude's Hospital in Memphis, Tennessee, every patient gets a pharmacogenomic consult. Now, those are high stakes, high risk patients sick with cancers and such, but every patient gets a test. there are also health systems across the US that are freestanding health systems where all of their patients get a pharmacogenomic test, like where they're standing service, just like any other service in a health system. You get a consult, you can just flag it. You have someone that reads the results and help you interpret them, and is already available. So in some places, the investment was made early, and those places are light years ahead of the rest of places that are limping along one person at a time trying to start, services. But broadly, it's hard to tell. I ran out of time. I wanted to do this exercise for this lecture to go back and see how long it took for blood pressure measurements to go from the invention of the device into being sold on the over the counter in stores, right? That might give us an estimate of how much time it's going to take. But I ran out of time. Like I just did. I ran out of time. Yeah.
Elvin, great talk, man. Thanks so much.
Thank you.
Yeah, a couple of questions. So, when people are hearing about 23andMe going under and your genomic data might be at risk, or police using reports from Ancestry to track down criminals and all of that. So with that scenario, I can see challenges in selling pharmacogenetics to patients.
Yeah.
Is there like some kind of a language that we use, because we need to teach our students this to counter this argument if they want the patients to benefit, right?
Sure.
What are your thoughts on that?
That's a great point. And there are risks associated with your genomic data being anywhere. There are risks. So there was this argument about if the data is de-identified somewhere, then it can't be unde-identified. And scientists proved that fact to be false as an academic exercise, right? You can find the results out there saying you can't. There's some risk associated with your genomics being tested by public companies. There's some inherent risk. So patients appreciate it when you are honest and open about those risks, and then they can make an informed decision, right? So even with, people will sign up for anything and do all types of take cutting edge risk with genetics if you're honest with them up front. So that's been my experience. Yes, sir.
I'm seeing a lot of those. Sorry. One thing I'm seeing also too is with the genetic testing is people trying to find their family's ancestry and whatnot. I'm kind of seeing it being used more for that than the medical side. So I've done the Ancestry 23andMe thing. And I get these emails all the time that, hey, do you know who this person is? Let me find this person's dad, blah, blah, blah. And I was like, oh, let me call Uncle so-and-so, see what he knows. You know what I mean? So, yeah, I think that's going to drive it more than the medical side, actually.
I think it's much more exciting than the medical side. But yes, Roscoe.
So I'll just talk louder. It seems that the uptake is low. Now a much softer voice. It seems that the uptake, by the way, thank you for a great presentation. This is like benchmark of what a good presentation is. So I'm just honored to be here and I've watched it. It seems that the uptake of pharmacogenomic testing is relatively low versus the value it provides. And I couldn't help but to notice that one of the diseases or types of diseases for which pharmacogenomic testing guidelines was surprisingly low is oncology.
They're actually high there.
But in one of the graphs, it was...
Yeah, so I didn't include oncology in my graph because the markers in oncology are extremely high, but they're not classic cytochrome P450. They're not classic PKPD markers, but the uptake there is much higher than everywhere else. And pharmacogenomics in oncology has helped drive much better success in drug discovery over the last 15 years.
But that was exactly my point.
Yeah.
Because in oncology, most products are, or most assets are actually sold under a companion or with a companion diagnostic. And so, which brings me to the point of uptake, where it seems that here the onus of pharmacogenomic testing is on the payers, on the patient side. But realistically, pharmacogenomic testing can be a way to screen the correct patients for which, or the correct patients that would benefit from the drugs that we manufacturers are marketing.
Yes.
And so, do you think uptake would change dramatically if regulatory framework said, no, 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 no. If you have this information available, you have to market it with a companion diagnosis or diagnostic testing to then make sure that you were not putting patients at risk by exposing them to drugs that they wouldn't respond to or they would respond adversely to?
That's a great question and comment. So about 15 years ago, you started to see, so about 20 years ago, drug companies were under the opinion that splitting their potential population was going to hurt their bottom line. So even though some of those companies were collecting genomic information in their studies, like early as starting studies in 97, like I know some where they were collecting the DNA in 97, right? And then did nothing with it, right? Because they did not want to split their potential population, but they were underestimating the impact of having someone have a toxic response and you losing your entire population over a drug, like harming 0.00001% of the population. So about 15 years ago, you started to see companies start these genomic groups within industry. And so you see some people working in industry now in genomics because they understand that if they can eliminate the risk of having those terrible, toxic adverse events, people dying, drugs being taken off the market, they win much more than if they just tried to keep their entire population. Now, in the early days of industry starting to incorporate pharmacogenomics, that relationship between industry and the FDA was maybe a little bit more contentious. Like they were not trusting that information would hurt them if they shared it with FDA. So the FDA created a voluntary submission program where if you have a drug company and you're developing a drug, you can voluntarily give that data to the FDA. Their scientists will help you work through it and evaluate, look for associations and such. And you see that they have co-authored papers between FDA and drug companies now where they're sharing that data with the world, and it's not to harm their potential, bottom line with developing drugs. So they're working synergistically now, and that has been helpful. So if in the Alzheimer's space, right, we've had these 3 antibodies developed in the last few years, like I'm saying few, like last 3-4 years, where the first one was associated with abnormalities on scans afterwards, right? Then the next 2 drugs that were developed after that, they actually include the gene that's associated with this increased risk of abnormalities in the brains of the people who were receiving these antibodies. So rather than lose the entire opportunity of getting their drugs on the market, they identified a subgroup that has the risk up front. It's a black box warning, and they got approvals. And okay, I won't say what I think about the drugs, but they're approved. All right, so that's a great question and point, Vasco.